Very good morning to you all. Today we shall continue with the cell. Today we shall discuss about eukaryotic cell. Eukaryotic means the nucleus is well defined, having nuclear membrane, nucleus and nucleoplasm. Beside this, membrane-bound organelles like endoplasmic reticulum, mitochondria, Golgi bodies, etc. are present. So we shall discuss the various components of eukaryotic cell. Where they are found? They are found in almost all the plant cells and animal cells. Clear? And protists as well as fungi group. Now, the first component that we shall be discussing today, that is cell wall. It is found only in the plant cells. That is the main difference between the plant cell and animal cell. Beside this, they are also found in fungi and certain bacteria. Now, when we talk about the plant cell, chemically they are made up of cellulose. Fungi, it is made up of chitin, and bacteria we have already discussed earlier, that is peptidoglycan. Clear? Now, what is the cell wall composed of? It is composed of three parts, that is middle lamella, primary cell wall and secondary cell wall. Middle lamella, it is the structure of the cell wall which provides rigidity because of the deposition of calcium and magnesium peptide. Beside this, you have the primary cell wall that is formed inner to the middle lamella. They are found in the meristematic tissues and parenchymatous cells. Secondary cell wall, how they are formed? When there is more depositions of the cellulose in the form of cellulose microfibrils, that is called as secondary cell wall. Clear? Now, secondary cell wall, that is this cellulose microfibrils, sometimes they are found in groups or clusters. Then they are called as macrofibrils. Beside this, there are two structures that may be found in the cell wall, that is plasmodesmata and pits. What are plasmodesmata? These are cytoplasmic strands. These are living cytoplasmic strands that joins two adjacent cells. Since cell wall is dead and is freely permeable, so how the exchange of materials can take place between the cells? It is because of the presence of plasmodesmata, which are fine cytoplasmic strands that join and help in the transport of materials between the neighboring cells. Beside this, there are in the vascular tissues, in the tracheids, xylem and vessels, there are thickened areas due to the deposition of lignin interrupted with unthickened areas which are called as pits. Clear? They provide mechanical strength as well as cell-to-cell interaction. Main function of the cell wall is to provide shape, that since it is dead cell, it allows the freely movement of the substances into and out the cell, and also they provide protection. Let me show you the diagram, just a brief diagram. That is, suppose this is a plant cell. Clear? Now, in this case, this is the middle lamella, and this will be the primary cell wall. This will be the secondary cell wall. Primary cell wall. I'll draw the diagram at the end of the session. Next, we come to plasma membrane. It is a living membrane, dynamic membrane. Clear? It is the structure found both in plant cell and animal cell. It surrounds the cytoplasm. There were many models proposed by different scientists. One was Davely Dennison in 1935. Next, it was Robertson who gave the unit membrane model or the concept, and lastly the Singer and Nicholson model, also known as fluid mosaic model, proposed in 1972. Clear? Now, what do we find? That is why it was all rejected. Davison's proposed that the membrane is made up of three layers, that is the protein layer outside and the lipid layer in between. Clear? Unit membrane concept, what does it say? It also proposes the same thing. That is, there is lipid bilayer in between with the protein layer on either side. But the protein layer is rigid. That was later on discouraged because when high resolution electron microscope was invented and the plasma membrane was studied, it was proposed that is the cell membrane or the plasma membrane shows the fluid mosaic nature, that is the quasi-fluid nature of lipids. Clear? Now quasi-fluid nature of lipids means what? That is the lipid layer shows fluidity. Clear? Having a hydrophobic ends and hydrophilic ends. The hydrophilic ends are water loving while the hydrophobic are water repellent. The protein molecules are on the either side. Clear? Now the protein molecules, that is the proteins, are regarded as globules that move in and out through the lipid bilayer. That explains the fluidity of the lipid bilayer or quasi-fluid nature. That is, the plasma membrane can undergo dynamic changes. Beside this, there are two proteins, intrinsic proteins and extrinsic proteins. Intrinsic proteins means internal proteins. They are tightly attached to the surface and they are held tightly to the lipid bilayer. Got it? And when we talk about the extrinsic proteins, they are also called as external proteins or extrinsic proteins. They are superficially attached to the membrane. Clear? Now beside these proteins, there are various types. Some are carrier proteins. What are carrier proteins? They help in the transport of materials in and out the cell. Some proteins behave as receptor proteins. They bind to the surface of the cell membrane and then they help in the transmission of information like hormones. Clear? Or enzymes. Some are transmembrane proteins. They are so large that they can cross the lipid bilayer and help in the transport of materials. Besides, they are also called as channel proteins. Clear? They also help in the transport of materials. Then you have the porins. These are large proteins which allow the smaller proteins to pass through it. Aquaproteins, what do? They attract or connect with any molecule outside the plasma membrane. They attach to it and then rotate and push that outside protein or the molecule inside the cell. Clear? That's why they are called as aquaporins. So how this transport of molecules or materials takes place? That takes place because of two types of transport, passive transport and active transport. So today we shall discuss about passive transport. There are two types, simple diffusion and facilitated diffusion. Diffusion, movement across along the concentration gradient from a region of higher concentration to a region of lower concentration. Now what do you mean by facilitated diffusion? Facilitated diffusion means you have the polar molecules which are hydrophilic in nature, so they cannot cross the lipid bilayer. Clear? So in that case, the carrier proteins, they get attached to this polar molecules and then they help in the transport to cross across the lipid bilayer and reach the cytoplasm. Clear? That is facilitated diffusion. So that takes place in three ways. That is symport, antiport and uniport. Symport means involves two molecules. When two molecules, they move across the membrane in the same direction. Clear? Suppose this is the membrane, two molecules moves along the same direction, that is symport. And antiport means what? That is, there are two molecules, one will move in this direction, the other will move in the opposite direction. Lastly is the uniport, That is, the movement of molecules can take place in any direction, independent of the other. Clear? So let me go through, give you the notes, so that will be more clear to you all. And I will, like last class, you had the flagellum. I had already given 3 parts, just this one. Then you have page 3, page nuclear flagellum. This is about the prokaryotic cell, which you are seeing continuation of the this one. Next come to eukaryotic cell. Then you have the plasma membrane. Beside this, I had discussed about the special structures found in the cell wall, plasmodesmata and pits. So plasmodesmata brings about connection. That means it connects two adjacent cells, or you can say one the cytoplasm of one cell with the other. They are the only living component present in the cell wall. Whereas the pits, they are found in the vascular tissue of, that is the xylem. One more page is there. According to your syllabus, you have to lay more emphasis on the fluid mosaic model proposed by Singer and Nicholson. You may be asked like this type, like, what do you mean by the quasi-fluid nature of the cell membrane or the lipids? The proteins appear as globules embedded in the lipid bilayer, just like a mosaic. That's why it is referred as mosaic, fluid mosaic pattern. Whereas the lipid are made up of phospholipids, oligosaccharides, water, and one more is little left, this much only. Diagram. Can you go through the diagram? Not this diagram, from the book. this diagram. Just see. One more diagram. One more diagram. Just a minute. That's all. Any spaces? One more diagram you can give. I leave it.